Okay, I think it's about time. We'll go ahead and get started. Everybody's here ready for the program, it looks like. So thank you all for coming. I'm John Kennington, President, and I welcome all of our guests here tonight. Our first thing we'll usually do is our recorder's report with some interesting birds that have been around lately. And Paul reports there was an Iceland gull, which is a form of the Thayer's gull on the River Parks Trail. And I'll tell you, spring migrants are starting to show up, but some early ones were an eastern kingbird at Keystone. Some sandhill cranes flew over Keystone at the eagle property we have. There's an eared grebe at Lake Ehola, prairie falcon at Lynn Lane, a white-eyed vireo at Oxley, and a Swainson's thrush. So those are some of the a little earlier than usual birds that have started showing up. Let me see here, so let me look at my... And as I mentioned, for those who maybe came in late, please congratulate Jim and Anita Mitchell on their 50th wedding anniversary. So, and there's a cake for them back there, so everybody has to have a piece of cake before you leave, because they do not want to take that home. And Glenn and Gail, who brought it, do not want to take it home either. So please have some cake. If you want to go back and get a piece now, feel free. Let's see, we're looking for, and one of our big events of the year is our backyard wildlife habitat garden tour in May. And this year it's going to feature 5 gardens basically in the South Broken Arrow area. And they've actually one in north Bixby, but it's very close to Broken Arrow there. So a nice convenient area. And Gail has a sign-up sheet for volunteers. So we need a lot of volunteers to help make the garden tour work. And this involves taking people's tickets and selling tickets. So we'll have a table with a couple of volunteers at each house. And it's Saturday. We basically have 2 shifts on Saturday: Saturday morning, Saturday afternoon, and then Sunday afternoon. And the big benefit is if you volunteer, you get a free ticket for the tour, which are, I believe, we're going to be $6. I think is what we've been doing, and we may raise that, but I think we're at $6. So, if you volunteer for 4 hours, you're making $1.25 an hour. So I think I don't know how anybody can resist. I mean, you may be turning people away, Gail. So. Would you remind us of the date? The date is, and I don't have it in front of me. 18th and 19th of May, Saturday and Sunday. So please, and Gail's you have the sign-up sheet here? Okay, it'll be on the back, or do you want to pass it around? Just leave it on the back. Okay, it'll be going around. So if you'd like to sign up to help with that.
I'll be checking with people about what time you want to work, what garden you want to be at, if you want to be with a specific vendor.
Yeah, you don't need to decide on a specific date or the time, right now. Just if you're interested, Gail will be in touch with you then. So thank you, Gail. And thank you, Aileen, for organizing this again this year. Flycatcher Trail, which is our garden out at Jenks High School, our model garden where we sort of demonstrate how you can grow native plants in your own yard. There's going to be a work day on the last Saturday of the month, March 30th at 9 o'clock. So if you'd like to do a little gardening out there, we could always use some help out at Flycatcher Trail. We also Tulsa Audubon is sponsoring a field trip. This is a long-distance field trip to Panama. So Jeff Cox, do you want to just talk about that for a second? You can keep. Oh.
If you're a member of Tulsa Audubon, get the emails or the Facebook, you've seen that we are sponsoring a 9-day trip to Panama, Canopy Family, Canopy Tower, and Canopy Lodge, the end of September and early October. Right now, we don't have enough people to make the trip. So if you've been thinking about it and trying to decide If you're really interested, please see me after the meeting. If this is the first you've heard about it and are interested, see me after the meeting. I forgot to bring the flyers this evening, but I'll be around and you can just come talk to me in person. So, but if we don't get enough people in the next month or so, you hear that?
Yeah, okay. Okay. And our next field trip is, on a little more local level, is next Tuesday on March 26th. It's an after-work field trip out at Lake Yohola. And, I don't have my calendar. I forgot to bring calendars. I printed them off and forgot that they're sitting on my printer at home. But check our website and on Facebook for the time. I think it's 4:30 is when we're meeting. And just for an hour or two, we'll bird around Lake Yohola, maybe go over into Oxley a little bit, but just very, beginning. You don't need to be an expert birder by any means for any of our trips, but especially for this one. You're welcome to come join us for that. Gail, you bring refreshments tonight, or?
Actually, we had several people bring refreshments tonight. Brandy Blankenship, Shirley Jones, Julia Harris, and Karen Goodman brought refreshments for tonight. We appreciate that. I'm going to pass the sign-up sheet around for the next 2 months, 3 months actually. Well, yeah, 2 months. And if you would sign up, you don't have to bring a lot. Just bring 1 or 2 items, and I'll provide the plates and tablecloths and all that stuff. So you just need to bring your food. I'll call and remind you when it's your time. Thanks.
Okay. Good. Another sign-up sheet going around here. The other big thing that we have going is our Lesser Prairie Chicken Festival, which is it starts April 10th. We have the first event is a field trip up to Pawhuska for greater prairie chickens, and then that weekend we're out in Woodward looking at the lesser prairie chickens. So again, that's a longer trip. And we're almost full. Some of the trips are sold out. And so far we've got people from 24 different states signed up. I think we've got a total between all the events, we got 81 people so far signed up. We're like I said, we're about at capacity so we're gonna have a really good year. And one of the reasons is our keynote speaker is gonna be Ken Kaufman. And I'm sure the birders in here, if you're a birder, you know who Ken Kaufman is, and even the non-birders probably don't, because he's also written one of the best butterfly guides, field guides to butterflies. And he's in numerous books and writes for Audubon and New York Times and just about, he's our current day Roger Tory Peterson, basically. So he'll be our speaker at the Prairie Chicken Festival, and he will be our speaker at the next Tulsa Audubon meeting. So he's going to hang around for a couple days after the Prairie Chicken Festival and be our speaker. So next month, Ken Kaufman. So if you are coming, I would advise to get here. Oh, we have some of the copies of his books back here too if you want to see some. So I would advise getting here, watch our website, Facebook page, and the emails for the exact details, because we'll have, probably before the meeting, have a book signing, and we will have some of his books for sale. He's got a new book about spring migration that's being published, I think, April 2nd or April 5th, and we will have copies of that book available for sale, so you can buy the book and have him sign it. So keep an eye out on our, all of our media to find out the exact details, but I would advise get here early, because we're going to, I just about guarantee will be a standing room only audience for that presentation next month here, April. Again, I don't know whatever this third Tuesday in April is. So 16th. Okay. So anyway, that's coming up in April. So keep an eye out, like I said, for information on Ken Kaufman being here in Tulsa. Are there any other announcements I'm forgetting? Oh, yes.
April 6th, we're going to caravan over to the Spavinaw area, and we will be doing birds and wildflowers, because this is wonderful native Ozark habitat. particularly in some of those Spavinaw areas which the city of Tulsa has owned since the 1920s, close to 100 years, and haven't really been disturbed. So we should see some good birds and good wildflowers if the weather cooperates. And the Indian Nations Audubon people, who are sitting right back there, a couple of them, are going to be going with us on that trip, and they know the area. And I do too, because I'm a former Indian Nations Audubon member. So that'll be a
Jim,
great
you want to
trip.
mention also our Pathfinder trip next month? Just a preview?
I don't remember what day we had that on. I've got it here.
Yeah. Well, anyway, we've got a trip coming
We've
up.
got a trip coming up for warblers to Pathfinder Parkway in Bartlesville, absolutely one of the best warbler spots in Oklahoma. And Mark Peterson, who is an expert birder with incredible hearing, will be going along helping out on that. I just know where to go. I can't see and can't hear, so I have to have somebody else along to help me find the birds. But Mark will be helping with that.
Okay, well, thank you. So we got
It'll be
some,
a great trip.
yeah, we got some good stuff coming up here the next couple months. Anything else I'm forgetting to mention? Oh, yeah, outreach. Thank you. Come on up.
We had a great day at the gathering place yesterday. They have special things during the spring break, and yesterday was Nature Day, and they had invited us to exhibit there along with Global Gardens, Master Gardeners, things like that. And we were the outstanding exhibitors. And I want to thank all of you who contributed at our year-end campaign for our outreach program. We spent a whopping $29 of it for materials on this. So but it makes a difference to have some money to work with. And we had 3 different games set up, and they had activities that they could do. Probably, you think at least 1,000 people came through there yesterday. It was from 9 to 3. And I want to thank Jim Dimming, Mary Jackson, Karen Goodman, where's Karen, who were there all day, and Nancy Hamill, and then one of our young birders, Jonathan Schneebs, who came in the afternoon and helped us. And so if you have any questions about what we did or, with the kids, I'll talk with you afterward then.
Thank you, Julia, for organizing that. Our outreach committee is doing some really good stuff. Got a number of programs we've given and developing all sorts of new good programs. Okay, if there's nothing else, then Cheryl, you want to introduce our speaker?
Our speaker this evening is Ana Chicas-Mosier, who is a doctoral candidate at Oklahoma State University. She has been researching the mechanisms and consequences of metal exposure in honeybees, and her dissertation has focused specifically on aluminum exposure and the immediate effects on caloric decision-making, movement, and neurochemistry in honeybees. Her work has shown that decision-making, movement, lifespan, and acetylcholinesterase are affected by exposure. And in addition to her dissertation work, Ana enjoys working with members of the community to discuss what paths can and should be taken to alleviate stress on both our native and definitely economically beneficial pollinators. So please welcome Ana.
Hello. Can everyone hear me? We're good? Okay. So as she said, my name is Ana Chicas-Mosier. I work with honeybees specifically, but I'm going to talk a little bit about our native pollinators, how both honeybees and our native pollinators are impacted by anthropogenic effects, by what we do, how we can change our activities to help reduce stress on these organisms, and then I'll talk a little bit about what my research is specifically with aluminum. So this is a USDA chart about covering about the last 10 years. We have acceptable winter loss, which is determined by a survey of beekeepers. And you can see that that ticks upward, as well as total annual loss, which is the orange bars, as well as total winter loss. So last year, we were really hopeful that our total annual loss had gone down, but recently, or in the 2017-2018 year, we resumed at a 40% loss of honeybee colonies. So that's almost half of the honeybee colonies, that's just in the United States. So we're losing a dramatic number of hives, and it's not just in winter. So as you can see in the older data, we were just looking at winter loss, assuming that was where everything was happening. But we're seeing honeybee loss even in the summer. We're seeing about 10% additional loss during non-winter seasons. So this is significant. It also is significant because beekeepers are becoming more and more accustomed to it. This really implies that this is impacting people's livelihoods, in addition to we're losing our pollinators. So that's that 21% of acceptable loss, which is about half of what we're seeing. So honeybees, they're the species that I study, but they're actually non-native and an invasive species in the US. They are from the Mediterranean. They were brought over in the 1600s by Europeans, and they weren't good for native species, much like the Europeans. They brought over many pathogens that hit the native species pretty hard. And we continue to see this. We continue to see pathogen transfer between honeybees and our native species. And I'll talk more about that in a minute. But they're valuable. We have a multi-billion-dollar industry in this country and in Europe, where they are not native, but closer to native, multi-billion-dollar industry, not just from pollination services, but also from direct produce that they're providing. So we can't exactly say, well, it's an invasive species, let's get rid of it. This is industry, this is economics. We rely on these species. So their decline is hitting us in our pocketbooks in addition to reducing the amount of pollination services that are available. But they're also susceptible to colony collapse. These little red dots on this honeybee are not hers. These are mites. So this is basically a honeybee tick called Varroa. I'll talk a little bit more about them, but they occur across honeybee hives. This is a global infestation. Most honeybees hives can survive some level of Varroa infestation, but it can become extremely harmful. So it's not just the honeybees. This is a relative abundance globally of bombus species, which are bumblebees. Our black bars are the 20th century, and then we have 2 years of more present data, although it is older data. And what we see is that in most bombus or bumblebee species, a huge decline between, and this is relative abundance, so we aren't just counting the number of bees that are available. This is relative to space and time. That we're seeing a huge drop. Got an insect. We're seeing a huge drop in most of our bombus species as well. So these are not just beneficial pollinators, but they're also charismatic. People enjoy seeing bumblebees just like they enjoy seeing monarchs. So this sort of drop is becoming noticed outside of science communities and noticed by just laymen. People are noticing that insect decline is occurring. The windshield effect is considered, if you think about your windshield 50 years ago, there were bugs everywhere, right? You couldn't drive down the highway without running through bugs. Now you go 60 miles and you're still relatively bug-free. That is bad. We need these insects for our ecosystems. Some of these species are seeing as much as a 96% loss, and that's in bumblebee species. This is a very significant value. So we're seeing more decline than acceptable, and that's due to 3 primary causes: habitat fragmentation, pathogens, and chemical application. And I'm gonna kind of break them down a little bit and discuss what each of them are. So habitat fragmentation and food limitation. So we have our plot of land. We can call it an acre for simplicity's sake. And we have wildflower patches. Assume that these are more than just one giant sunflower head. They're patches of flowers. And then we have our hive. And this could be a solitary beehive, or I've made a picture that makes it look like a honeybee hive, because that's what I do. But we add homes to it. We cut out those nice patches of flowers. We then add roads. We add more homes. Now we have 3 of those patches. We've basically eliminated their food source, and the food sources that were here, now they have to travel much farther. That's a lot more of an energetic cost for them. So this is costly to our bees, whether they're solitary, native, honeybees, whatever. So now we're going to couple that with pathogens and diseases. So we're already having food stress. Now we have varroa, which I kind of talked about. It's a larval mite. So these varroa are hitching a ride on this adult female, but they grow, and their kind of bread and butter is in the larval state. And they prefer the drones, which are male bees. They represent a very small proportion of the hive, but they're bigger, therefore there's a higher amount of hemolymph or blood. There's better nutrition in a smaller or a larger space. More varroa mites can fit on a drone. So as I said, many hives have some level of infestation. We also have nosema, which is an intestinal fungus, and kind of paradoxically, it is aided by the use of fungicides. So when bees ingest fungicides that we've applied to corn or to our yards or whatever, it actually increases the probability that nosema is occurring in their guts. So this is a graphic of what nosema looks like. So this is Nosema apis, which is basically the honeybee version of nosema. This is Nosema ceranae, which is the bumblebee version of nosema. Can you guys see a difference there? They're a little bit longer, they're a little rounder, but what about this blended guy? So this pathogen that was originally found in Apis mellifera, or honeybees, is actually transferring and mutating to infect our bumblebee and solitary bee species. So our application of fungicides is not only affecting this nosema level in Apis mellifera, but also in these integral species diverse native pollinators. We also have, and this is going to be specific to honeybees again, foul brood, which is a larval bacteria that causes death in the larva. It causes the hives to smell horribly. It is the smell of death. And recently, there have been dogs that have been trained to seek out the smell of foul brood, because once an apiary has a hive with foul brood, it's so highly contagious that if you don't catch it almost immediately, the whole apiary can be wiped out in a matter of weeks. So finding it quickly is extremely important. And one of the frequent ways to get rid of an infected hive is to burn the whole thing, because it is that infectious and that horrible to your apiary or to your livelihood. Also chalk brood, which is a larval fungus that basically makes the larva look like chalk. These 3 are extremely deadly. The bottom 2 are extremely contagious. So we're working to try and figure out ways to remediate these effects, but also as we compound these things, food loss, pathogens, now we're going to talk about chemical application, all of these stressors on the bees are going to compound to create a much harder environment for them, much more difficult. Whereas if they just were fighting with maybe nosema, they would have a greater chance of survival. So chemical application, Neonicotinoids, I'm sure you've all, yes. So they cause foraging deficits, motor function decline, olfactory, which is scent reduction, and they have been recently in 2018 banned by the European Commission. So neonics in Europe are basically not a thing, but we continue in the US to use them. We continue to not block their usage. The EPA has been kind of dragging their feet on this. So they're still affecting our pollinators. They're likely still affecting other organisms, but they're still in effect. Again, fungicides, increasing that concentration of nosema, that interaction between the two. And then my area of research are metals. So I specifically look at aluminum toxicity. But when we cause acidification, we emit carbon dioxide into the atmosphere, bringing down acid rain, changes soil pH. Metals that are traditionally in the soil and are not hazardous can become hazardous. And I'm finding that with aluminum, and I'll talk more about this later, that it is causing cholinergic damage. Other metals can also cause misfolded proteins and many other effects. So how can we help to alleviate decline? I'm going to kind of break those three down again and offer solutions for all three. So we have habitat fragmentation. This is the same graphic that I already showed you. We can plant more native species. That's pretty easy. I'm sure most of you are doing that already if you're trying to attract hummingbirds or other bird species into your yards, right? So if you're planting native species, you're bringing in more native pollinators, and increasing our species diversity of native pollinators also helps to continue to grow these native plants. Because although honeybees are my area of interest, they aren't the most efficient pollinators. We can also, and this is my kind of graphic of doing so, let your lawns grow. So we live in an economy of manicured lawns in the United States. You trim your lawns, you put sod down of non-native plants that are not drought hardy, but that also isn't good for our pollinators. Letting the clover and the dandelions stay a little bit longer. The clover right now is thriving, and so are the bees are loving it. If you watch a clover patch right now, there's tons of bees in the middle of the day. Not in the early morning because it's still a little too cold for them. So how can we help with the pathogens part of it? Reduce bee movement. So this is a 18-wheeler. Each of these boxes is a hive. If this hive is sick, everybody's sick. They're all squished in together for, sometimes these trucks are going from Florida to California for almond groves. This is a huge transfer of potential pathogens. And I'm sure you've seen pictures of these. Actually, not too long ago, one of these fell over and there were just bees all over the highway. So there's other effects of bee transfer other than pathogen transfer, but it's overall most likely not the best practice. Additionally, as I mentioned, those pathogens are switching from honeybees to other species. If we're moving bees from Florida to California, we're also moving those pathogens to native bee species that exist in California but may not exist in Florida. Minimize immunosuppressant use. That's the fungicides and the nosema, the gut flora. Provide clean, fresh water during summer, and I'll talk more about this. If, just like the birds, having a bird bath for bees is helpful. They get hot in the winter or in the summer and need to cool down not only their own bodies, but also the hive, and I'll talk a little bit more about how they do that. Chemical application, basically don't do it. Minimize it. If you need to put down Roundup, that's something that you need to. At least read and follow the labels. We have a lot of problems with people not diluting chemicals properly, and this is going to affect all species, birds, bees, what have you, more drastically than the research is going to identify, because the research is based on the chemical labels. And then reducing carbon emissions. You drive less acid rain, less soil acidification, fewer metals. All of these things are interconnected, and all of them contribute to our loss of species in general, but in my case, specific to pollinators. So, wow, now I have two. So climate and bees. I kind of talked about this. We release emissions into the sky, whether it be industry or vehicular. That comes down as acid precipitation. Acid precipitation can it's affected boreal forests in Canada. It's affected bees, and I'll talk more about that. But also pathogens thrive in warmer weather. Varroa is going to do better in a warmer climate. Nosema is going to do better in a warmer climate. So although the bees it may be easier for the bees to survive, it's also easier for their pathogens to expand in their numbers. And it may be getting too warm for solitary bees, or some solitary bees. A lot of them live in the dry arid areas of the southwestern United States. As we continue to warm, those areas are going to become even hotter. Phoenix and its melting runways. This is not good for solitary bees. Additionally, aluminum, like I mentioned. So toxicants do not have to be directly lethal to be harmful. Aluminum is likely a sublethal effect, but if it's affecting the motor or the olfactory sensitivity of a honeybee, she's not going to be able to forage properly. She's not going to be able to find the best food source. She's not going to be able to provide for her hive. And if the foraging members of the hive aren't bringing enough food to the neonates of the hive, then the hive can't survive well. So what can you guys do? Specifically, you guys. And I went to the National Audubon Society to try and figure out, well, what are they suggesting the best DIY projects for birds are, and how can I apply that to bees? You guys they need similar things. That's super easy for me. Bee baths. So I'm sure many of you have bird baths so that you can get nice birds in the summer months. If you basically apply the same thing to the ground and add some rocks or some twigs, the bees need something to land on. Otherwise they can, so this isn't exactly the best example, although they could land on these flowers.
Wine corks.
What?
Wine corks.
Wine corks, exactly, something floatable, so that they have a source of water that they can land on and that they can retrieve. If it's a renewable source of water too, some of the apiaries that I've been to just have a hose on some twigs 24 hours a day during the summer. So the bees have a constant stream of water. That is for like 50 hives, but it's still really beneficial to these insects. Bee nesting boxes. Have you guys heard of these?
Oh, yeah.
Okay, so they've been kind of popular right now. It's as easy as getting a piece of wood and drilling some holes in it. A solitary bee, and this Acustom allows for different sizes of bee. So we have sweat bees that are teeny tiny. They look like ants, but with wings. Or we have huge carpenter bees, right? So allowing for different sizes of bees, what solitary bees do is they'll lay an egg in the back of one of these, and then they'll pack it with pollen and saliva, and then they'll lay another egg. So they'll do that all within about 48 hours, so that they're hatching about the same time, and then they all exit at the same time. So solitary bees don't have parental care in the same way that honeybees do, but they are providing their larvae with a food source. And then once they exit the hive, then they're solitary. But they also come in all shapes and sizes. This is a much larger version if you really want to help save the bees. But this can also be used by other organisms as well, lizards, sometimes bats, so that it's basically a small homemade home for native species. Plant native species. Tons of websites offer native Oklahoma flowering species. Planting those in your yard, not cutting down the ones that occur in your yard, provide food sources to the bees. The best things to do for native species is to plant native plant species, because those are the most nutritional pollen and nectar sources for them. Something like a dandelion or a sunflower, they can collect from it and they can eat it, but it's not very nutritional. It's kind of like eating candy all the time. So having a variety of native species is helpful. Dandelions. The other thing the National Audubon Society suggested is to leave your brush piles in the winter to provide a home or a hiding place for birds. But this also provides underneath a place for solitary ground nesting bees. And if these twigs were larger, it also provides a place for larger wood nesting bees. So these also help. This isn't just bee specific. Insects love these, and insect communities really build them up. Don't use Roundup. Roundup's bad for birds. Roundup's bad for bees. It's marketed and is still for sale in the U.S., but glyphosate is increasingly studied as having negative effects on our ecosystems. So now I'm going to shift gears. I've talked about what's wrong, how you guys can help it. Now I'm going to talk about what I do. So I don't actually have my own hive. I do a lot of international research looking at different subspecies of honeybee, and I use a variety of different methodological means to answer questions about how aluminum is realistically affecting honeybees. So this is a picture of a free flight experiment. So she is completely free to return to her hive, to come to my flower patch. She's not harnessed in any way. She's got a little white dot on her bum to identify her as white abdomen. And then we can track, where is she going? Blue or white flower? These are flowers. It tricks them. And we can determine, how does her color preference change with sucrose? So and I'll talk more about this, high quality reward, you should stick to that flower color. And they have amazing vision, so they can tell the difference between blue and white. This is a cap pushing experiment, and I'll talk more about it. This bee is given 2 flower color options and has been trained to push one of these caps to receive a reward. So one contains water, one contains sugar, and you'll see a video of that. This is what we call a monitor system. Each of these tubes can house a bee, and it monitors the bee every minute for as long as she's alive. So we get a lot of movement data, we get survivalship data, we get how well she's able to maintain a day-night cycle. So is she staying 12-hour movement, 12-hour no movement, or is she kind of moving throughout the day? And then we can use neurochemical assays. So this is what a honeybee brain looks like. These are the 2 eyes. These huge triangles are all of the optic nerves. That's how well their sight is, or how well connected their eyes are. These are what we call the mushroom bodies. That's where most of the brain activity occurs. And that's what we dissect when we do brain sections. Yes, you can dissect a brain out of a honeybee head. It is tedious, and you fly brains everywhere, every time. So I'm going to talk first about the brain assay that I did. This one I didn't have to dissect heads, thankfully. It's a modified element assay. It's colorimetric, so basically the brighter the color, the more protein in her brain. So I expect that aluminum is going to, oh, this is an American bee. The honeybee subspecies aren't differentiatable by their looks morphologically, but they are genetically different and they behave differently. So to distinguish them on this presentation, this will be Apis mellifera mellifera, also known as the American bee. She's actually a she's actually a European subspecies because they're invasive here, but they're typically what we have here. So this is a cholinergic enzyme. I'm gonna go a little technical here, so get ready. We have our presynaptic neuron. We have acetylcholine into the synaptic space being received by the postsynaptic neuron and causing excitation. This is what causes you to do this. It also allows you to remember things. This is all the cholinergic system. And it's conserved from bees all the way to us. So this system allows us to move, allows us to remember. It allows bees to move. It allows bees to remember. So it's very important. When we expose bees, or just about any organism, aluminum blocks this acetylcholinesterase, which is a degradation enzyme. So it's basically what allows acetylcholine to be taken up and to stop this excitation. So that's when I go, and I didn't continue moving. But without that reuptake, I just keep going. And eventually, I'm going to get tired, and my cells are going to start dying. And eventually, that's going to cause paralysis, and eventually, I'm going to die. So this is bad. We don't want this. But what I see is what's called a hormetic effect. So low doses actually cause the greatest inhibition of acetylcholinesterase. And I'll show you what a graph of what this looks like here in a second. So it's dose dependent and possibly hormetic. It's basically a U-shaped curve. High doses resemble controls. Again, American bee. So this is that curve. It's kind of small, but the highest doses cause the most drastic drop. We then see that in activity. So they are moving more. They are doing this when that inhibition occurs. So that's super exciting. This was like I was jumping around the room and dancing because my data match. So enzyme activity translates to daily motility. It also reduced their survival. So bees that were exposed to aluminum, no matter what the concentration, 10 milligrams per liter. I don't know if I said this, but in pollen concentrations, we've seen as high as 300 milligrams per liter. So this is very small comparatively. And they lived on average about 10 days less. That's a big impact on survival. These bees aren't maintaining their ability to stay alive. So significantly reduced captive lifespan. So this is that flower patch experiment that I was talking about. So basically, if a bee has all the same sugar, we expect her to act correspondingly. All flowers, doesn't matter. But if blue flowers contain a higher concentration of sugar, we expect her to act accordingly and to just visit blue flowers. As well as the reverse. White flowers contain the high reward. She visits white flowers, right? But what we found is that, finish her little dance there, exposed bees couldn't maintain this. They reduced their ability to pick the correct color. So bees that were exposed, and this is at about 2 milligrams per liter. So 300 in pollen, 2 is causing this effect. So this is not good. This is that cap pushing. So we have a yellow cap and we have a blue cap. Again, this is an American bee. Oh, this is a Turkish bee. Let's hope. There she goes. So this is 8 times how fast she actually does it. So the yellow cap didn't contain sugar. Blue cap did. So she's drinking. Does anyone want to see it again? I'm just going to do it one more time. Yellow cap, you can see it moving. She pushes it. Finds the sugar. So she is actively problem solving on this. And we did train her to push this cap with incremental movements, but she problem solved. But we found with Puerto Rican bees that they demonstrate a distinct yellow preference. You don't give them aluminum, you just give them sugar. They have a preference for yellow. Which likely means that at the time that we were collecting the data in Puerto Rico, there were a higher abundance of yellow flowers. So they had learned to visit yellow. But when you give them aluminum, they abandon their preference and stop visiting yellow hardly at all. They start visiting blue. So that is changing their ability to adapt to changing floral environments. So realistically, if exposed to aluminum in northern or eastern Canada or Brazil or Puerto Rico, likely the preference is going to be abandoned. And they need these preferences.
So Anna,
Yes?
how do bees get exposed to aluminum just in a general environment?
Thank you. I meant to mention that. So in the general environment, we have typical soil, right? When soil acidification events occur, such as bauxite mining, which is the extraction of aluminum, or acid rain, when the soil pH reduces past about 5.5, ionized aluminum becomes available. So usually aluminum is tightly bound in aluminum hydroxide. It can't be really taken up easily. And even if it is, it's excreted fairly quickly. But when it's in its ionized form, it can be mistakenly used for similar elements such as zinc and magnesium, which are micronutrients, which are needed. So it can be taken up into plant roots and then distributed in pollen. And in Brazil, in bauxite mined areas, that's where they found the highest concentrations of 268 parts per million, which is, like I've said, I haven't, other than in the motility, the movement and survivalship data, all of the free flight has been up to about 20 parts per million, and I've found effects. So factors, tons of factors, and they're compounding, and a lot of them we can do small things to try and remediate or to try and alleviate, but because these factors are all compounded, it's difficult to disentangle what is the most strong impact on bee decline. But we can do our part. And humans are not limited to our direct effects. Anthropogenic effects are all over the place. It's driving your car. It's planting bees, or planting flowers, not planting bees. It's everything, every small decision that you make as a human can affect the environment positively or negatively. So being aware of how things are interconnected and how things are interacting with our environment is important. And then, now I'm done.